Now that we have an understanding of some of the different types of neurons, we're going to dive into nerve impulses or action potentials. So to start us off, a nerve impulse is simply a very quick message. And it's a very quick message that is sent in a particular part of the body, being the nerve fibre. Now, as we mentioned, they're very quick, and what this allows for is your body's quick response to stimuli, compared to other systems, like the endocrine system, which leads to quite a slow but long-lasting change. The nerve impulses themselves are what we refer to as an electrochemical change. And this is because it is a change, firstly, in the electric voltage of the cell, both relative to the inside and outside of that cell. And secondly, it's brought about by a change to some of the concentrations of ions found inside and outside of the cell. So looking at a change in particularly sodium and potassium. The very quick physics lesson that we'll do here is all about potential. Now, you may have heard before that like charges repel and opposite charges attract. And when we're talking about this, the reason that we bring it up is an electrical force is what pulls opposite charges together. And this force increases, as those charges get either closer together or the charges themselves get larger. When they come together, there is energy that is released. And this leads us to separate these charges, or if this leads us to separate those charges, they then have the potential. It doesn't mean that they will, but it means that they have the potential to come back together, and we can call this the potential difference. Now, this potential difference is very important for us when we're talking about this electrochemical message. And what we do is we measure it in millivolts. And millivolts, as you'll represent in your responses throughout the year, will be lowercase m, capital V. So, action potentials themselves. If we think of the cell membrane as a fluid environment that is surrounded by a number of different ions, an action potential is a change in the concentration of ions on either side of the cell, both in the extracellular solution and in the intracellular solution or the cytoplasm. So this diagram here is a full depiction from a start at a resting state all the way through to returning to that resting state after a period of rapid change. So what is an action potential? An action potential itself is a rapid depolarization and repolarization of the membrane, and they only last roughly about a millisecond. Okay, so again, they're very, very quick. A nerve impulse, by definition then, ever so slightly different, is the movement of that action potential down a nerve fibre. Okay, so two distinct terms here, both equally important. So what we're going to dive into now is the action potential before we can talk about the movement of that action potential down the nerve fibre. So we have a few key steps. First step, the cell at rest. At rest, the extracellular fluid contains a high concentration of sodium ions. So this is in the extracellular fluid, meaning outside of the cell. And in this diagram, you know what, I'll make it nice and simple for you. We'll use the blue highlighter because that's what sodium is represented as. In our extracellular solution, you can see all of these sodium ions. Lots and lots of sodium outside. There's a little bit of sodium inside as well, all right, just a tiny amount in the intracellular fluid. On the inverse side of things, if I get an orange in favour of a red highlighter, we can see that there is a limited amount of potassium outside the cell, but there is a larger amount of potassium inside the cell. Now, both sodium and potassium have a charge of +1. Okay, they have a charge of +1. They have the same charge. But what is also found inside the cell is a number of large negatively charged proteins. And I'll highlight these large negatively charged proteins in green. And we can see that they are only found in the intracellular fluid. This means that at rest, the cell tends to be more negative inside relative to the outside of the cell. And this is A, because there's more sodium outside the cell than potassium inside the cell, and also due to the presence of these negative or large negatively charged proteins. This is the cell at rest. When we talk about the resting membrane potential, and this comes down to our potential energy that we were talking about, or potential difference that we were talking about earlier, this is because, again, there are more sodium, keep to blue, sodium outside the cell than potassium inside the cell. And what this means is it creates a gradient of charge. And we've measured this charge, and it is found, and it's important to note that the resting membrane potential, so I'll write it over on the left here as well, is negative 70 millivolts. When a neuron itself has the negative membrane potential, it is said to be polarized. So right now, this membrane is polarized. It is resting. Now, the concentration of sodium is about 10 times greater inside the neuron than out, okay? The concentration of potassium is 30 times greater inside than out, okay? So when we're talking about sodium and potassium being positively charged, there's simply not enough potassium to counteract the effect that sodium is going to have on the cell, and also to counteract the effect of the large number of negative ions found inside the cell. So I'm going to skip over this diagram very quickly now, and we will talk about what happens to start this action potential off. So again, our cell is at rest, and in stage 1, what we've just been talking about, the cell is at negative 70 millivolts. Now, a stimulus, meaning a change, causes some of the sodium channels to open. And some of these sodium channels opening leads to sodium ions entering the cell, because they flow down their concentration gradient from being in a high concentration outside the cell to a lower concentration inside of the cell. As they move, they bring their positive charge, and that positive charge results in the inside of the cell becoming more positive. Okay. So, what does this mean? It means that this negative 70 is going to begin to increase. It increases, again, as more sodium ions enter the cell. Now, second step, depolarization. If a sufficiently strong stimulus is applied, so a sufficient change, then enough sodium enters the cell to reach this line here. This line is known as the threshold. And this threshold is negative 55 millivolts, so 15 millivolts higher than the resting membrane potential. If this threshold is met, all of the sodium channels in the membrane, at that particular region of the membrane anyway, will open up, and that causes a rapid influx of sodium ions into the cell. And this is why we can now see a rapid increase. If I highlight this one here, you can see the rapid increase in that charge inside the cell, okay? Because that charge increases very quickly as the inside of the cell is now more positive than the outside of the cell. When we look at the next step, okay, eventually our cell has this large difference, and we've reached about positive 30 millivolts, as I've highlighted in yellow at the top here. We move on to the third step, known as repolarization. And during repolarization, all of these sodium channels are closed. Sodium has flooded in, and so the cell has tried to close those channels. Now, in order to try and balance this charge, what it does is it opens potassium channels. So a key part here is that potassium channels open, and they are in a higher concentration inside the cell, so they move from inside the cell to the outside of the cell. And what this means is that, as we can see in our graph over on the right, the charge decreases. Okay, that membrane potential returns, and it quickly returns to a negative number as that electrical gradient is re-established and the cell membrane itself is, keyword here for our stage, repolarized. But the potassium channels stay open for slightly too long. And as a consequence of this, we end up in the fourth stage, known as hyperpolarization. Because too many potassium ions left the cell, okay, this means that the membrane potential drops down roughly to about negative 80 millivolts. As a consequence, there's a special structure found within the cell membrane known as the sodium-potassium pump. And the role of this sodium-potassium pump is to re-establish the membrane potential or the resting membrane potential. The sodium-potassium pump wants to re-establish the resting membrane potential. And it does this by moving 3 sodium out and bringing 2 potassium in. So a slightly unequal distribution. As this occurs, we can see, firstly, 3 sodium being regulated by the sodium-potassium pump, and we can see 2 potassium being brought back in. This here is the action of the sodium-potassium pump. Now, after a period of time, this is eventually going to, if I scroll back up here for a second, return the membrane to that resting membrane potential, so negative 70 millivolts, and it is this time that another action potential may be triggered. So, when we talk about action potentials, there's some important things worth noting. Number 1, when we talk about the threshold, okay, this needs to be met. If the threshold is set, and it always will be set for us at negative 55 millivolts, the cell difference must reach this. So sodium is going to gradually flood into the cell. Once it reaches negative 55, then depolarization will occur. Okay? If it does not reach negative 55, then no action potential is generated. Okay? And this is what we call a graded potential. Okay? So then the membrane, instead of kicking up, the membrane instead is going to kick up a little bit, not reach 55, and then come back down back to negative 70 until it can have a much larger response and reach that negative 55 millivolts. So when we talk about a threshold, it is what we need in order to generate an action potential. Okay. So this here is another representation, a good diagram to see this action potential, and it's labelled as we go as well. So we've alluded to this one here previously when we said that we can't undergo another action potential during hyperpolarization, but we can actually extend that period out. Because when the cell has undergone depolarization, it can't undergo another set or another action potential until it returns to that resting membrane potential of negative 70 millivolts. So during that time, again, it cannot undergo another action potential, and what that helps with is preventing signals from travelling both directions down an axon. This is sometimes also known as the refractory period, so the period in between 2 action potentials. So lastly, what we've got here, if we start over on the left-hand side, so this is a nerve cell found in a finger, and in this nerve cell we can see that sodium is gradually diffusing into the nerve ending to create depolarization, and that depolarization then will lead to the action potential itself. So over on the left, when sodium diffuses into the nerve ending, this is what we can refer to as stimulus. It's going to lead to a change in the membrane potential, triggering that action potential.